Excuse me. Okay, class. So, good afternoon. Now, this afternoon, class, we're going to discuss your Module 1 Introduction to Immunology and Serology. Now, as much as I wanted to upload the original recording, there were just too many nonsense, and as well as plenty of discussions that weren't really much directed or related to it. So, as to preserve and allow a better video recording, I'm just gonna make a new one. Now, the first module class would be all about your introduction to immunology and serology. Now, as you all recall, immunology class would refer to your resistance to disease, specifically to infectious disease. Now, it consists of the following: the study of the molecules, cells, organs, and systems responsible for the recognition and disposal of foreign non self material. Now, here, class, an example of molecules involved in immunology class are your antibodies. While for the cells class, we have your WBCs. For your organs, it could be the skin, it could be the lymph, lymph nodes. It could also be your spleen, and in some cases, even your thymus. Now, another organ system involved here, class, one of the most important and the major is your lymphatic system, which we will tackle in your module 2. Now, it is also the study of how body components respond and interact, the study of the desirable and undesirable consequences of immune reaction. So, what would happen, class, if there is the interaction between your antigen and antibody? Then it is also the study of the ways in which the immune system can be advantageously manipulated to protect against or to treat disease. So these are the composition of your immunology class. Then let's talk about the history of immunology. Now we have here Thucydides. Now Thucydides class was a Greek scholar. Now he was a Greek scholar class wherein it was believed that the concept of immunity from disease would date back to at least in the 5th century BC. So that it was at that earliest stage that there was already the concept of immunity. Now, he recorded individuals who contracted the plague at that time, and those who recovered would become immune or exempt. Now, if you recall, class, your plague would be caused by the bacteria Yersinia pestis. Now, it was believed, according to Thucydides, that if you would get infected by Yersinia pestis through the infected rat mites or rat lice, and you get bitten and you would somehow recover, class, you would become immune or exempt. However, class, the earliest recognized attempt to intentionally induce immunity to an infectious disease was in the 10th century in China, where they practice what you call variolation. Now, variolation, class, is the exposure of a healthy people to material from the lesions caused by the disease, either through putting it under the skin or inserting powdered scabs from smallpox pustules in the nose. Now, variolation, class, during that time would usually involve the disease smallpox. Now, smallpox, class, is similar with your chickenpox in its appearance. They would produce what you call pustules. Now, itong mga pustules na to, class, these pustules, wherein in variolations, class, they would get a part of the pustule and they would put it under the skin, or they would insert the scabs of the pustule into the nose. So imagine how disgusting that was. So again, in variolation class, it would usually involve the disease smallpox, wherein they would get the pustules or the scabs of the smallpox and introduce it to a healthy person by putting it under the skin or by introducing it through the nose. So like this class. This was done in China before. Then let's talk about one of the pioneers in variolation. We have here Lady Mary Wortley Montagu. Now, si Miss Montagu class, she was the British ambassador. She was the British ambassador's wife in Turkey. Now, during that time class, she found out about variolation, and when she returned back to England, she promoted variolation and had her surgeon variolate her 4-year-old daughter in the presence of the king's physician. Now, due to that class na amazed, that the king's physician was amazed. Now, and that led to the surgeon Charles Maitland to perform what came to be known as the royal experiment. Now, this royal experiment class would usually involve 6 prisoners. Now, this 6 prisoners class would be variolated. They would be introduced to smallpox. And fortunately, these prisoners would later survive. And that started class, the introduction of your variolation in England. Now, a better mode of variolation was introduced by Edward Jenner, and wherein he inoculated James Phipps with material obtained from a cowpox lesion that appeared on the hand of a dairymaid. Now, class, cowpox is the bovine version of your smallpox. Now, they would usually be found in the udders, UDDERS, of cows. Now, what happened here, class, was that he inoculated an experimental subject with smallpox without producing the disease. Although this experiment justifiably lacked an appropriate control, further studies by Jenner established the efficacy of his vaccination procedure. So imagine that class that with the help of Jenner, vaccination, this was a form of vaccination during that time, wherein he inoculated cowpox lesions onto a person named James Phipps. And after that class, he would inoculate again a smallpox. And what happened was that James Phipps became immune. So basically, the vaccine here that he used was the milder cowpox. And due to the exposure of the cowpox class to James Phipps, that James Phipps became immune to the more aggressive form of which is your smallpox. So I hope you understand on how that became a vaccination method. So this is your smallpox class, usually found in the skin of humans. And here we have your others or your cowpox. So ito yung others class wherein the milk would pass through. So Edward Jenner took uh, cowpox lesions inoculated to James Phipps. Then he waited 6 weeks. After 6 weeks, he inoculated smallpox naman. And because of that, being inoculated first with cowpox, James Phipps now immune, became immune to smallpox. Now, here's the difference between variolation and vaccination. So, variolation would actually use smallpox virus to protect the people, while vaccination by Jenner would use the far less dangerous cowpox virus. So, among the two, of course, we would choose vaccination, which is less troubling compared to your vaccination, compared to your variolation. Then we also have your Louis Pasteur class. Now, Louis Pasteur class observed by chance that older bacterial cultures would not cause disease in chickens. Subsequent injection of more virulent organisms had no effect on the birds that had been previously exposed to older culture. Okay, what he did here, class, was that he tested for your chicken cholera. Now, chicken cholera, class, was caused by your Pasteurella multocida. Now, what he did here was he took old cultures, gave it to the chicken, treated it, treated it, then it became healthy. Then he introduced new cultures, but the chicken would now become immune. Again, what he did here, class, was that he tested for chicken cholera, which is caused by your Pasteurella multocida. What Louis Pasteur did was that he introduced old cultures and gave it to the chicken. The chicken would now become diseased, and that after that he would treat it. Then he would introduce a more virulent strain, but this time the chicken would not be affected or would become immune to it. In this manner, class, the first attenuated vaccine was discovered. This event can be considered the birth of your immunology. Now, the word attenuated here would refer to the weakened form. So weakened because it used an older form of the bacterial culture. Then significant milestones in immunology. Now, in 798 class, Edward Jenner discovered your smallpox vaccination via the use of cowpox. Then in 1880 to 1881, Louis Pasteur would use live attenuated chicken cholera and anthrax vaccines. 1883 to 1905, LMH Tikov would develop the cellular theory of immunity through phagocytosis. So we have your neutrophils and your monocytes that would involve in phagocytosis. Then in 1890, Emil von Behring and Shibasaburo Kitasato developed the humoral theory of immunity. So when you say humoral, class, this would usually involve your antibodies, which will be discussed in a more depth in the coming modules. Then in 1891, Robert Koch demonstrate the cutaneous or the delayed type of hypersensitivity, which can be a more deadly type because it could lead to difficulty in breathing. Now, he also identified class your Cox bacilli, also known as your MTB. Then, in 1894, Jules Bordet discovered complement. So, complement classes are a group of proteins. A module is made for this one. Then, in 1897, Robert Koch identified precipitation. Then, 1900, Paul Ehrlich identified the antibody formation theory. 1902, Charles Richet and Paul Potier made the discovery of immediate hypersensitivity and your anaphylaxis. Then in 1903, Maurice Artus developed the Artus reaction of your intermediate hypersensitivity. 1903, Wright and Douglas developed opsonization. 1938, John Richardson Marak developed hypothesis of antigen antibody binding lattice formation. 1944, Medawar developed the immunologic process in transplantation. So in transplantation, class, you have to consider that you just can't receive any random organ or random tissue from a stranger. There has to be a compatibility when it comes to certain factors, which we will be discussing in the coming lectures. Then in 1949, Jonas Salk and Albert Bruce Sabin developed your polio vaccine. Now, Salk and Sabin class, you have to just remember that because the name of your polio vaccine would either be the Salk vaccine, or the Sabin vaccine. So the vaccine was named behind the two discoverers for polio vaccine. Then in 1951, Walter Reed discovered the vaccine against yellow fever, and he also identified the graft versus host reaction. So this was the one I was talking to you about, class, your GVH, which could be seen in incompatible incompatible transplants. Then we also have 1957 Frank McFarlane Burnett for his clonal selection theory. 1964 to 68, we have the T cell and B cell cooperation in immune response. 1972, we have Gerald M. Edelman, Rodney R. Porter for the structure of antibodies. Then 1975, George Scholar and Cesar Milstein would develop the first monoclonal antibodies hybridoma technology. Then in 1980, we have Baruch Benacerraf, Jean Dosset, and George Snell, which would develop the major histocompatibility complex, which is now being used in transplants class. Then in 1983, Luc Montagnier discovered things about HIV. 84, Robert Gallo discovered also about HIV. 1985 to 1987 was the discovery of genes for T cell receptor. 1986 was for the discovery of your monoclonal hepatitis B vaccine. 1986 was wherein Tim Mossman discovered Th1 versus Th2 model of your T helper cell function. And in 1987 class, Susumu Tonegawa discovered antibody diversity. So the different types of antibodies. Then 1996 to 1998 was the identification of toll-like receptors. 2001, FOXP3, which is the gene directing regulatory T cell development. Then in 2005, Tracer would now develop the human papillomavirus. Now, your human papillomavirus is known to cause your cervical cancer and your warts. So he discovered the vaccine for HPV. Now let's talk about the definition of terms. Now, immunity class would refer to the resistance to infectious agents, foreign particles, toxins, living cells, and cancers. Now, there are two types of immunity. We have your active and passive immunity. Now, active immunity class would result from natural exposure to an infectious agent or administration of a vaccine. Now, in active immunity class, your body creates antibodies when exposed to foreign particles such as bacteria, viruses, fungi, and in some cases, it can also be done by exposing through vaccines. Again, active immunity class would basically, from the word active, your body is actively creating antibodies when exposed to foreign particles, such as your bacteria, viruses, and your fungi. And an artificial way to produce antibodies would be through vaccine. While passive immunity class is a type of immunity that results from the transfer of antibodies from the immunized host to a non-immune individual. Basically, class, from the word passive, there is no antibody production. Instead, there's what we call antibody introduction. So you would get antibody from another source. It could be from your toxoids and other matter that we will top discuss in the future topics. So that is your passive immunity. Again, passive immunity basically from the word passive. There is no action. There is no antibody production. Instead, antibody is introduced from another source outside of the body. Then we also have your adaptive immunity. Now, adaptive immunity class is a type of resistance that is characterized by a specificity for each individual pathogen or microbial agent and the ability to remember a prior exposure, which results in an increased response to that pathogen upon repeated exposure. Now, adaptive immunity class would usually involve your vaccines. Now, when your body gets infected, let's say by a virus, antibodies would be produced for that virus. Now, let's say you would get healed, you would get treated, okay ka na, magaling ka na, you're well. You get infected again by the same virus. Your memory, there would be the action of your memory class, memory of your body, wherein the same antibodies now would still fight the same viruses that entered your body. So that is your adaptive immunity. Again, your adaptive immunity class would allow your body to remember a prior exposure. It usually involves vaccines, wherein a certain pathogen, let's say a virus, would infect your body, and antibodies would be produced against that virus. Now, let's say you get treated, you're well. Now, with your body remembering the previous exposure to the virus and you get infected again, this time it would be milder because you already have the necessary antibodies to kill the virus immediately. So that's your adaptive immunity. Basically, from the word adaptive class, your body was able to change or to adapt to that kind of disease. And then we also have your innate. So when you say innate, these are natural or basically what your body already has. So this is the ability of the individual to resist infection by the means of normally present body functions. So this is how your body would normally fight any disease. So there would be the skin. Then we also have your WBCs. We also have your lymphatic organs. So this would allow your body to fight any infection. It just refers to what your body already has. Then we also have your antibodies class. Now your antibodies class would appear in a Y shape. Now these are produced by your B lymphocytes. So if you recall class, certain lymphocytes when triggered would become plasma cells. Your B lymphocytes or your B cells would become plasma cells and they would produce antibodies. Now this antibodies class would just respond to foreign substances exposure. Antibodies are also known as immunoglobulins. Now we also have here your antigen. Now antigen class is any macromolecule that is capable of eliciting formation of immunoglobulins or sensitized in cells in an immunocompetent host. So antigen class would be any foreign particle. It could be a virus, it could be a bacteria, and in some cases it could be a fungi. Now antigens would cause the production of your antibodies. Excuse me. Now we also have your affinity and your avidity class. Now affinity class would refer to the initial force of attraction that exists between a Fab site on an antibody and one epitope or a determinant site on the corresponding antigen. So Fab site class. Imagine this would be your antibody. This would be the antibody. Now the Fab site class would usually be at the tip of your antibodies. So this is the Fab site. Now, this Fab site class would allow your antibody to attach to any antigen. Then we also have your epitope or your determinant site. Your epitope class is a part of the antigen. It is the determinant site where the Fab would attach to the antigen. So this is your epitope. Yan ang epitope niya, or the determinant size. That's your affinity. Now, your avidity class would refer to a multivalent antibody which would bind to a multivalent antigen. So certain antibodies class would have more Fab sites. They would usually become, an example would be your IgM antibody, which is a pentamer, meaning five Fab sites. So one, two, three, four, five. As you can see, this is an example of your IgM. Now, due to the many Fab sites class, it would usually attach to many epitopes, as seen in the diagram. So that's the difference between your affinity. Affinity would just refer to a single attachment or a double attachment, while your avidity would refer to three or more attachments to the epitope or the determinant site. Then we also have your central tolerance class. Now, this would refer to the destruction of potentially self-reactive T and B cells as they are mature in either the thymus or the bone marrow. Now, class, we have two types of cells in your lymphocytes. You have your T cells and your B cells. So, T cells in class would usually develop in the thymus, while your B cells would usually be developed in the bone marrow. So, just remember the first letter. T cells, T, thymus. B cells, B, bone marrow. Just for you to remember the organ where they will mature. Now, what happens here, class, if there is overproduction? Overproduction of B cells and T cells, it would cause your body to destroy them in the thymus and in the bone marrow. So that's your central tolerance. Again, central tolerance class is the destruction of your B cells and T cells due to their overproduction or overreactivity that could lead to autoimmune disorders. They're already destroyed in the main organs palang class, in the thymus and the bone marrow. While your peripheral tolerance class would refer to the destruction or repression of your lymphocytes in the peripheral lymphoid organs that could respond to self-antigens. So let's say, class, that certain B cells and T cells were already released, but they are still overproduced and are overactive. Now, what happens are the side organs of your lymphatic system. The side organs of the lymphatic system, such as your lymph vessels, your lymph nodes in the spleen, would destroy the T cells and B cells to avoid, again, autoimmune disorders. So that's your peripheral tolerance. Just remember, class, that when it comes to central tolerance, it involves the major organs, thymus and bone marrow, while for peripheral tolerance, it would involve the side organs, lymph vessels, spleen, and lymph nodes. Then, let's talk about the epitope and the paratope. Now, the epitope class, as I've already discussed earlier, is also known as your Fab site. This is the key portion of the immunogen against which the immune response is also known as the determinant site. Okay, sorry, that's wrong. This is also known as Your epitope is also known as the determinant site. So this is the one found in your antigen. While your paratope is the one known class as your Fab. So it would usually attach to the determinant site. Then let's talk about the chains in your immunoglobulin. So we have here the light chain and the heavy chain. Now the light chain class is a small chain in an immunoglobulin that is bound to the larger chain by disulfide bonds. The two types of light chains are called your kappa and your lambda. While the heavy chain is one of the polypeptide units that makes up an immunoglobulin molecule. Each immunoglobulin monomer consists of two heavy chains paired of two light chains. So this is your immunoglobulin structure class. So it would usually appear with two pairs of heavy chain. Yan siya. So this is the heavy chain. Now, your light chain class would appear here. Let's use a better color. This one. This is your light chain. Now, they would usually be attached by disulfide bonds. So, yan sila. They would be attached by disulfide bonds. So, your light chain is known as either kappa, and the other one would be known as your lambda. So, here you could find your Fab site or your paratope, where they would attach to the epitope or the determinant site. Then we also have your Fag fragment and your FC fragment. Now, the Fag fragment class would refer to the fragment of an immunoglobulin molecule obtained by papain cleavage that consists of a light chain and one half of a heavy chain held together by a disulfide bond. So if you recall, di ba, yung itsura, the appearance of your immunoglobulin earlier. So this is the heavy chain and this would be the light chain. So the Fab fragment here is this one. They consist of a light chain and one half of a heavy chain. So this is your Fab fragment. And your FC fragment class is a fragment of an immunoglobulin molecule obtained by papain cleavage that consists of two carboxy terminal halves of two heavy chain. So this is your FC fragment. Let's go back to the slide for better understanding. So this is this one. This is the entire Fab fragment. Consists of one light and one heavy chain. Dito, 10 class. One half dito yung dulo. And then for the FC fragment, this entire part is the FC fragment which is composed of two heavy chains. So pag tinanong sa exam, what are the pair of your Fab fragment? It consists of one light chain and one heavy chain. Kapag FC fragment, both heavy chain. Then we also have your human leukocyte antigen, also known as your HLA. Now, this is a protein coded for by the human MHC genes that has essential role in the immune response and the rejection of foreign transplants. So we will discuss this class in a future model and is involved in the major histocompatibility complex. Then we also have the cluster of differentiation, also known as your CD. Now, these are cell surface molecules expressed on leukocytes and other cells relevant for your immune system. So one thing you need to remember about this class is about this T cell. They have markers CD4 and CD8. Now, si CD4 class is also known as your T helper cell. Si CD8 naman is your cytotoxic cytotoxic T cells. Helper cells, cytotoxic T cells. So we'll discuss this furthermore in the future topics. So that ends our module 1. So you will have your quiz next meeting. So please try to focus on more on the handouts, but you could also read the overview of the topic. So thank you. Goodbye.